was a fairly austere beginning, but I learned to love music. I didn't know much about science until I got to high school and had a chemistry teacher who got me really excited about science as a detective story where you can discover things. And that is something I guess I wanted to do. And because it was a chemistry class, I figured I should be a chemist. So I went to the University of Virginia and I majored in chemistry. I totally avoided the life sciences because they just seemed really messy. And I went off to get a PhD in quantum mechanics. And my PhD project was mostly computer coding, which in those days was very different than it is now. Trying to solve second order differential equations that were what you needed to do to understand collisions of atoms and molecules. But along the way, I realized I had been a little limited in my perspective, and I discovered there was this other chemical that was really interesting that I'd kind of ignored. This one, the double helix, DNA, this remarkable information molecule that is the instruction book for all living organisms, including us. And I had pretty much just skipped over paying attention to that. And all of a sudden it seemed like, wow, this feels like the center of the center. This is what I really want to work on. And without being quite sure what to do about that, I figured, well, let's keep all the options open and I'll just go to medical school. Now, that, mind you, was a bit of an odd decision to make by somebody who was already a couple of years into a PhD program in physical chemistry, but it was the best decision I made because I got to medicine and realized this is what I really am excited about. So I learned what I needed to learn about taking the care of patients as part of medical training and residency. And then I had to figure out how to do the research part. I ended up back at Yale learning those skills. And then finally, as an assistant professor at the University of Michigan. Now, this was not a linear pathway, and I don't recommend it to you if you're in a hurry to get to the point of being independent, but it's just sort of the way it worked out. And I think that's probably true for a lot of people. Things that you think are going to be straightforward oftentimes are not. So at Michigan, my job, as I set it up, was to try to find the causes of genetic disease by studying that DNA molecule and finding misspellings in it that might account for conditions like these. For instance, cystic fibrosis. And by the way, that little picture of me sitting in a haystack holding a needle was a picture I had made to try to explain to people why it was so hard back then to actually find the cause of a genetic disorder when you didn't really know what you were looking for. And the genome was 3 billion base pairs, and you were looking for something that might be just one of those base pairs that was out of place. But we did succeed with cystic fibrosis even before there was a genome project, and believe me, it was tough then. And Huntington's disease a couple of years later, and neurofibromatosis about the same time. It's actually a fairly common disorder, but it doesn't get much attention. More recently, I've been working on a disease called progeria, which is a cause of premature aging. That one also is caused by a single letter, and that's a T that should have been a C. My lab continues to work on that today because we now know not just the cause, but we have some pretty good ideas about what to do about it and an FDA-approved drug and are on the pathway towards maybe trying to cure this with gene editing. But then this happened. I was happy at Michigan thinking, okay, this is going to be what I do, teaching medical students, taking care of patients, running a research lab. But the Genome Project came along, and I was pretty excited about that. And the first leader, the guy on the right there, was none other than Jim Watson, as in Watson and Crick. And it was Bernadine Healy on the left, who was the NIH director at that point, who hired him. But that didn't go very well. Dr. Watson, for all of his brilliance and his recognition when he founded the halls of Congress, which was really helpful, could be, rather insulting. And after he called the NIH director a lunatic more than once, basically he was gone. And then I was getting a call because that's what apparently her search committee thought would be a good idea. Have a physician come who really loves this DNA stuff and see if he'd like to run the Genome Project. This was a shock. I was really happy where I was. I said, I'm so flattered. Being a federal employee is not part of my life plan. I think the answer is no. But she called back. And after a while, I realized, how can I say no to something that might be the most significant project ever undertaken in life science just because it's not convenient? So I decided to say yes. And I'm glad I did because it was an amazing ride. guess this is one of my other lessons maybe for you is when a door opens that you don't expect and it doesn't seem like really what you had in mind, well, look at it anyway and make sure this is not really an opportunity to do something even more exciting than what you had planned. So yeah, I could spend a long time talking about what happened over the next 10 years as we took those initial goals of the Human Genome Project, which seemed at the time as absolutely out of reach, and maybe we're just going to have to admit failure, but bit by bit, with lots of technology coming on board and lots of computer science as well, because you had to assemble the human genome from short stretches of DNA, usually 100, 200 base pairs, and then patch it all together using appropriate means to avoid making mistakes, because the repetitive sequences in the human genome could easily cause you to misidentify an overlap that wasn't really there. But we figured out how to do that, initially with simpler organisms and then moving on to the human genome. And so by 2000, we were able to say we had a draft, and by 2003, we had what we called at that time the complete human sequence, although it left out some of the messy parts in the centromeres and telomeres, which have only recently been completed. And so this was an experience of leading a project with 2,500 scientists in 6 countries. It was a pretty big, complicated enterprise, but I loved it. These were wonderful people. They basically didn't care too much ultimately about who got the credit. They just wanted to be part of this phenomenal historic undertaking of reading out our own instruction book. And we did. And it has been transformative. And probably most of you who are working in human biology are using all kinds of genome sequences and probably have a hard time imagining how we did anything before we had this information at the click of a mouse. Believe me, it was tough. And now it's still tough, but it's a whole lot easier. The goal next for me as a physician was to say, how are we going to use this information to advance the cause of medicine? Because at the point that we had the whole human genome, we couldn't really read it. Only about 2% of it coded for protein. The rest, who knows what's there? It must have something to do with how genes are turned on or off in the right times in the right cells, but we didn't understand how that would work. So a whole lot of other projects had to get started to try to fill in information about function. These are just a few of them. Privilege of having a chance to lead most of those for the sort of the next 5 years after the Genome Project sequence was done. But then I kind of got a little bit restless. I left NIH for a year, tried to write a book, got involved in being the science advisory committee for a candidate for president. That was Barack Obama. And then when he got elected, somewhat to my surprise, was then called to come back to NIH, this time as the director. That's Building 1, the headquarters. That was me on my first day going, oh my gosh, this is what I'm now supposed to do? Wow, really means I've got to be knowledgeable, not just about genomics, but about everything in biomedical research. I better surround myself with some really smart people who will help me understand things that I don't understand. And I better be careful about the kinds of decisions I make, that they're always going to be based on knowledge and evidence and not just some sort of gut feeling, because that's not a good way to go when you're trying to manage a scientific operation. So I did get a chance as NIH director over the next 12 years to do a bunch of things that were pretty exciting. Reorganize things a bit to start a new component of NIH called NCATS, the National Center for Advancing Translational Sciences, trying to make sure that all the basic science discoveries that were happening got moved forward into the clinic instead of just sort of sitting there, which sometimes was a bit of a problem. And that is a very exciting part of NIH now 10 years later. And I got to work on neuroscience, an area that I hadn't known a whole lot about, and to recognize that the time was right to really mount a very bold effort to see if we could understand how those 86 billion neurons between your ears do what they do. And this is the Brain Initiative, which is also now at about a 10-year period, and has been doing an amazing set of things to understand not just the cell census in the brain, although that's been pretty astounding, but also how these cells work together in circuits that enable us to do really astounding things. This is going to be transformative for our understanding of brain diseases. And I also got to spend time with leaders who, many of them were pretty interested in science. Here's one, for instance. This is a meeting in the Oval Office where we were talking about whether the time is right to actually take everything we're learning about genomics and really move it forward in what you could call precision health, where you really try to read out the genome sequences of lots and lots of people, and you understand how that interacts with the environment to tell whether somebody is going to be healthy or whether they're going to slip into a chronic disease that might have been preventable if we understood this in a better way than our one-size-fits-all approach. And this is Obama, who's leading the discussion and who always had the best questions in any science meeting that I got to go to. Not that he was a scientist, but he really understood how science works. And he was always thinking a step ahead of what it was that we were telling him about. And out of that came a project which is going on today called All of Us. I don't know if any of you are part of this, but you could be. It's not too late. We're still enrolling. This is aiming to enroll 1 million Americans in a prospective longitudinal long-term study where your genome gets sequenced, you answer a whole lot of questionnaires, your electronic health records are shared, and researchers have access to all that information, but they don't know it's you. It's all been anonymized. And already we're up to about 800,000 participants on the way to 1 million. And a lot of really cool discoveries are starting to happen with this kind of very powerful and very diverse resource. Half of the people in All of Us are from racial and ethnic minorities. So we have the chance really to look at health disparities and diversity in new ways that we haven't had the chance to do before. So there it was, things seemed to be going along, and then it was 2020, and then this happened. As the NIH director in January of 2020, it was clear that we were headed into a very dangerous place as this virus that was clearly causing a great deal of sickness and death in China was starting to spread across the world. And it was my job to try to be sure that all of the scientific energy and ideas and materials and resources that could be put together on this would do so. That meant bringing together people from public sector and private sector, trying to make sure that everybody was learning from everybody else as quickly as possible, and particularly the development of a vaccine that might protect people from what at one point by March or April was leading to 4,000 deaths per day in the US. Working with Tony Fauci, who reported to me as the head of the Infectious Disease Agency, we pulled out all the stops on the vaccine development and explained what we were doing to various leaders. Here you see from February 2020, Tony is telling President Trump about this. On the far left is Barney Graham, who is an amazing virologist who was leading a lot of the effort here. And on the right is Kizzmekia Corbett, a postdoc who was doing a lot of the most important work and is one of the heroes of this story. And so we did, in fact, as you may remember, push that forward. And by November, late November of 2020, the astoundingly positive results were unveiled that the mRNA vaccines, both of them, the Pfizer vaccine and the Moderna vaccine, had efficacy between 93 and 95%, which is astoundingly good for a vaccine. Most vaccine trials fail. I do think history will look back at this as one of the most amazing achievements of science ever. And here we were, ready to begin to scale it up and offer it to people who needed it. And that then was the focus. About a year from this, the focus was distribution, a different president in the very same space in the lab of the Vaccine Research Center on the NIH campus in Bethesda, Maryland, hearing about what this vaccine was all about and what we needed to do to get it out there. And we did. You were a part of this, I'm sure. If you looked at those curves of total vaccine doses being administered starting in January when they were approved by FDA, you got a pretty good slope there. But you got to about May, and then the slope didn't keep going up, and it started to go down. And unfortunately, that wasn't because everybody had been vaccinated who really needed to at that point. This is where we ran into what I had not anticipated, which was that there was a lot of misinformation, a lot of skepticism about whether these vaccines were really safe and effective. Even though the hard data said so, there was a lot of things that were circulating in social media and sometimes being promoted by various politicians that made people worry about whether this was something they wanted to take advantage of. And the saddest and most tragic part of that is that over the course of the next 9 months or so, from June of 21 to March of 22, there was a whole lot of unnecessary COVID infections and about 230,000 unnecessary COVID deaths from those 50 million people who basically said, I don't trust these vaccines. I got to tell all of you, this is something that troubles me deeply. I didn't see this coming. I thought that we were all mostly rational actors, which, when presented with data about something, would make a decision that was good for ourselves and our family. But in this case, that wasn't always happening. And there was so much of the misinformation that good, honorable people were distracted from what might have otherwise been the truth and attached themselves to other kinds of rumors that led them to believe, I just don't think I want to do this. So this is a case where our societal malaise, which is considerable, are having shifted off into divided particular corners, has led not just to some unfortunate conversations, but actually to people losing their lives. We seem to have lost some of the traditional anchors that we've depended on for our society to be stable and to have a healthy future going forward. One of those is the very issue about truth. I hope none of you would think that truth is just your opinion when it's something that's an established fact. But increasingly, you may be hearing statements like, well, that might be true for you, but it's not true for me, which is fine if you're talking about your opinion about whether a movie. It's not fine when it's talking about whether data that you could trust or not. If the data is there and it was a double-blind randomized trial with all the appropriate controls, that's truth. But that wasn't the way it was always seen. So we have a problem. Truth has become much more subjective. People feel comfortable saying, well, I don't like that fact, so I don't have to believe it. Facts don't care how you feel. Facts just information, reliable information from somebody that we don't like. But we are ready to embrace information from somebody who's in our own bubble, who may actually not be reliable. We've gotten that all tangled up. And trust in everything seems to be slipping. So I'm telling you all this in the middle of this talk because I feel pretty strongly that this is an issue for all of us. We are, as scientists, in a place of having access to truth about how things really work. It's going to be more and more important for us to be ready to explain that. Not in an aggressive, in your face way, but in a way that basically shares what we as people with special access to special kinds of information have some responsibility to share with others. And I wrote about that in a book that just came out a month ago called The Road to Wisdom on Truth, Science, Faith, and Trust. So that was a little distraction, but an important one. And this was driven in my mind by seeing what happened with COVID and realizing how much trouble we are in because of our divisiveness, which of course, given the way in which we've had months now of electioneering, which has only increased our divisiveness, you can't help but look at where we are and say, we got to pull ourselves out of this. And the politicians are not going to help. We're going to have to do this ourselves. Okay, enough of that little distraction. On to some other ideas about sort of digital science. And let me just tell you a little bit about my own research lab. This is my group at NIH, standing out in front of our research building, Building 50, also called the Lewis Stokes Laboratories. That group that you see there includes a couple of postdocs. It includes some senior scientists who are long-term stable parts of the enterprise. It includes a lot of trainees, and several of those in this picture, 5 of them, in fact, are post-bacs who have finished their college degrees and are trying to figure out what they want to do next. Maybe go to medical school, maybe go to graduate school, maybe neither of the above. And they come to NIH for a year or two and they get a really good experience, I think, being immersed in cutting edge science to sort of see how that feels. So I'm very fortunate to have a terrific group of people that I work with every day. And in the lab, there are 2 major projects. One, I mentioned already, this study of this disease called progeria. But the one I want to briefly tell you about, because it's really more in the computational space, is genomics and epigenomics of type 2 diabetes. Type 2 diabetes is an extremely complicated disorder if you're trying to sort out the genetics. We know it runs in families, but it certainly doesn't follow any recessive or dominant or X-linked rules. It's polygenic. And we used to think, well, maybe that means there's 10 or 12 genes involved. Turns out to be a bit more complicated than that. How do we sort it out? Well, once the genome project was done, the next step was to get a catalog of variations in the human genome. And we now know a very good catalog, although we need to do a better job of sampling people who are not European, because a lot of the work was initially done there. And that makes it possible to do a GWAS, a genome-wide association study, where you can collect DNA samples from controls who don't have the disease and cases who do. If you're looking at a disease, you can also do this for a trait, like a value of cholesterol, for instance. And then you genotype all of those DNA samples at all of those variable places in the genome, where maybe I have a T and you have a C. And then you look to see, is there a skewing of the distribution of those variations for the people who have the disease versus the people who don't? And that means a lot of genotype data and a lot of crunching of that data and cleaning of it so that you're not being misled by errors. And then some statistics to say, okay, it does look like this particular variant is happening more often in a case than a control, but what's the statistical evidence for that? We have to be pretty stringent about that. And then you end up with something at the bottom where you have the chromosomes aligned the way that we often do. We call that a Manhattan plot because the peaks kind of look like the skyline of Manhattan. So we started this in serious efforts once the Genome Project had finished and HapMap had produced a beginning map of variation. And the very first success of this effort was in about 2008. And at that point, we were pretty excited because we found like 10 places in the genome. Well, how's that looking now? This is the most recent publication. Up to 611 loci, segments of DNA in the genome where there's a variation that adds to your risk of type 2 diabetes. And you can see from the numbers that includes almost 500,000 cases and 2 million controls. So you get more and more power that way. Most of these variations have a very modest effect on risk. Might increase your risk by 1 or 2%. But if you have this many people, you can even discover those with a very stringent p-value of 5 times 10 to the minus 8. My graduate student, Henry Taylor, is co-first author on this paper. And this is probably not the end of the story because there'll be another one sometime. And it would not surprise me if we get up to 1,000 loci. So that's all very cool, but does it really tell you what's going on? This is where things get pretty woolly because you might have expected, and I think naively many of us did, that these variations were going to be in the coding region of genes. They were going to change an amino acid or affect a splice or something. No. 95% of these loci that cause this kind of increased risk are not in the middle of a gene. Some of them are quite distant from any genes, but they must therefore be part of the regulatory apparatus. That means they're really hard to work on. Quickly show you what you have to do if you're going to take that GWAS finding and actually get to a story about a gene. First of all, we have this nasty feature, although it actually has turned out to be helpful, in the human genome, that variations that are right next to each other tend to travel together. What's called linkage disequilibrium. And that means if you get a signal where it looks like, okay, there's something here that's predisposing to diabetes, it's probably not going to be that one variant and that's it. If you go to the next one over, the one after that, they're all traveling together, kind of in lockstep in what you call a haplotype, which means then you have a set of possible contributors, and one of them is probably causative, and the rest are along for the ride. Statistical geneticists, using a whole lot of inputs, break that down into what's called a credible set. So maybe there is like 72 different variants that are all traveling together. Some of those, it turns out by various math analyses, are probably not the culprit, but a certain group are still reasonable, and you end up with that credible set, each of which has some sort of possibility of being the right one, but it doesn't tell you what the right one is. Then you got to do some experimental work. And you do that, and then you try to figure out, okay, I now know this is the causative variant, but it's still in the middle of nowhere. So what gene is it actually affecting? That's what you want to know, the effector gene. And there's a whole bunch of experimental and some computational approaches to figure that out. That's what a lot of us are doing these days in the genetics of common disease. I'm talking about diabetes. This same diagram would apply to schizophrenia or to Alzheimer's disease or to Crohn's disease or to macular degeneration, but basically any human condition you can think of that's not Mendelian has this kind of inheritance and is being pursued in this way. So we're doing all of that, but we also want to bring it to the reality of how does this actually happen in a disease like diabetes, which means you also have to get into the nitty-gritty of which parts of the body are most engaged in whatever it is that goes wrong in this metabolic disorder. And here in this simple diagram are tissues that we know are all important. You can't just say, well, it's one thing. It's all of these things. Although it's fair to say that the islets in the pancreas that make insulin, when you really look at all the places where diabetes seems to be happening, that's probably the most important. But others are happening too. We had a great lab meeting today on new insights about the hypothalamus in the brain that clearly plays a role in predisposition to diabetes because it controls appetite. The problem is you can't biopsy somebody's hypothalamus, at least they won't generally appreciate that. And you can't actually biopsy their pancreas either. So if you really want to study places like that, you would feel like you're kind of blocked from getting the insights that you'd like. Except now, with this whole concept of induced pluripotent stem cells, you can actually begin to do those things. So you could take a skin biopsy from one of the patients in your study, or even a blood sample. With appropriate addition of the right 4 genes, you can convince those cells to become basically pluripotent. And then for the pancreas part of this, in a very carefully worked out recipe, you can convince those iPSCs over there on the left, over the space of, oh my gosh, 33 days, to go to the point of traveling through the same steps they do, or they did in each of us, because we all started out as one cell, and make ultimately at the end of those 33 days, a beta cell, which is the cell in the islets of the pancreas that makes insulin. It's pretty astounding that one can do this. You can also start with that same iPS cell and convince it in just 28 days to become an arcuate neuron in the hypothalamus. So we have windows into tissues that we previously couldn't see. And it's pretty elegant. This is an example of what, after 33 days, you can generate a 3-dimensional clump of cells that all started out with an iPS cell, which before that came from the skin biopsy of a patient. And here they are, stained on the left with insulin in green and a transcription factor called NKX 6.1 in red. On the right, it's the same islet now stained with insulin in green and glucagon in purple. So this is a lot like a pancreatic islet that makes both glucagon and insulin in separate cells, right there growing in your lab dish. Of course, this means, because now we have this system, that if you want to understand how a particular gene plays a role in this development and might therefore predispose somebody to diabetes as an adult, we have a system where we can manipulate the genetics in order to see this. This is a very complicated diagram, and I won't try to walk you through it other than to say that's what's done in this particular paper that we just published recently, taking that same system and knocking out a total of 12 different genes and 12 different experiments. You can really scale this up now. Sorry, it's not 12, it's 20 different genes. And each one of them asking what happened to the ultimate development of those clumps of cells that look a lot like islets. Did it happen or not? And the number of genes that are scored here, the ones that are in those red histograms, those are places where the knockout of a gene really did serious damage to what you would hope would happen with an islet. And those are all genes that we know were involved in diabetes risk. So we're kind of pulling this all together. None of this would be possible without an awful lot of computational approach. If you walk in my lab today, you would see 2/3 of the people are on the computer and 1/3 are at the bench. And that's very different than it was 10 years ago. We're more and more in that space. And that's true, I think, of most labs that are doing genomics. So let me try to wind this up here in terms of some conclusions or some broader insights, if I have any to offer you. When people come to me saying, oh, I know you were the NIH director for those 12 years, and I'm just trying to figure out, I'm just getting started in this area of biomedical research. What should I work on? What are you most excited about? Well, I'll give you a quick list. And it's very much a personal list. And do not be upset if your own favorite passion is not here, because this list could go on for hundreds of elements and it would not be extreme at all. But I am excited about neuroscience, that brain initiative, figuring out how the brain works. Woo, that's going to be a revolutionary idea coming forward. I'm very excited about what I just mentioned, the idea that you can do single cell biology and ask a cell what it's doing. Look at its RNA expression, look at its chromatin structure, That's brand new in the last 4 or 5 years. And it just completely changes everything in our understanding of life science. I'm excited about stem cell research. Those iPS cells I just told you about just give us chances to do things that I would not have dreamed would have been possible 15 years ago. I'm excited about gene therapy and the opportunity to use things like CRISPR and gene editing, not just to sort of look at things in the lab, but even potentially to do things to help patients, like we have now been able to do with the curing of sickle cell disease with gene editing and with gene therapy. With cancer, I'm particularly excited about bringing together what we've learned about genomics and the immune system. And I don't think it's crazy to imagine in the next 10 years or so that we will be able to figure out how somebody with stage 4 solid tumor metastasis could still be cured. That's the big challenge right now. I'm very excited about AI. I spent several days in Silicon Valley a couple of weeks ago. It's pretty clear most of the really top-notch AI folks in Silicon Valley don't know squat about life science, and they're thinking about other applications. And if we can figure out how to bring these fields together even more effectively than we have so far, I think some interesting things are going to happen, and they already are with things like imaging. I'm excited about this precision health idea that we don't have to do things one size fits all, both in prevention and treatment. And I'm somebody who believes that we're all part of one family, and I've spent a lot of my time trying to nurture global health right now in a big project on trying to build research capacity in Africa. And I think that's part of our responsibility also, living in a country that has all these resources. And I guess ultimately, I'm excited because I'm a doc and I care about alleviating suffering and trying to cure disease, that all of this feeds into that space. So I will defend that list, but I will also say there's lots of other things that could be here and just didn't happen to pop up when I was making the slide. Finally, I often get asked, what have you learned along the way? And particularly for people who are earlier in their own journey, what lessons would you want to share? I didn't keep my horizons wide open at the beginning. I told you that story of getting all excited about physics and chemistry and completely ignoring life sciences for a long time. That wasn't healthy. Eventually, it turned out all right. But I wish I had stuck with the same idea that science is interesting from all kinds of perspectives. There's no part of it that you could say, oh, well, that's boring. But oftentimes, maybe because we feel a little bit like, oh, gosh, I got to be an expert in something, you narrow down your view and you stop paying attention to what else is going on. So don't make that mistake. Look at that, that issue of Science Magazine. What are the hot topics that are being published this week from all over science? Maybe there's something that will pique your interest, because oftentimes the things that are most exciting and productive is when two fields come together that hadn't previously been talking to each other. That means also nurturing your curiosity. There's nothing about how nature works that's not interesting. Nurture that and go poke around and see what's there. I don't think I have to tell this group about coding, but I tell every other trainee that I have the chance to work with, if you haven't learned to code, you better learn now, because otherwise you're going to be screened out of an awful lot of interesting things. That doesn't mean everybody has to be a software developer, but it does mean you need to be ready to write a script when you need one and not wait for somebody else to help you who might not really understand the question you're trying to solve. And you need to find mentors who can advise you, and mentors like to be asked. So don't be shy when you're in the middle of something and you encounter somebody who seems like they know something about that and they're interested in your project. Say, can you be a mentor for me on this? And I think most of the time you'll get a happy smile and a positive response. Don't feel like you can't do that. Don't feel like it's only the people who are assigned as your supervisors that you can ask for that kind of advice. I mentioned this about the doors, like the door that opened for me for the Human Genome Project. And I initially said, no, not that one. But I'm really glad I took another look. So check it out. Sometimes it might not be what you expected, but it might be something even better than what you expected. And sometimes the doors close. And that can really hurt when it was something you thought was just right for you and then it didn't pan out. But sometimes that's also just as well. When you're at a point where you have a chance to pick projects, then pick something really important. A lot of the time in your training experience, you kind of get assigned a project by your mentor. So maybe you can't do a whole lot about that. But when it gets to be your opportunity, you could pick a project that's sort of the obvious next step in a well-trod area and you'll make a small addition of knowledge in that space that will add a little bit, epsilon, to human information, and that's good, and you'll get to publish a paper, and there's nothing wrong with that. But if you're going to spend your whole life in science, boy, it seems a lot more exciting if you can pick something that's a bit more profound than that, which often means it's going to be a little more risky. So that means you got to be willing to take some of those risks. Don't be afraid of that. As you saw from me, certainly don't expect to travel a linear career pathway, because I don't think anybody does anymore. And be cheerful about it. It's an amazing time to be a scientist. And yeah, there's all kinds of political and societal problems that I've mentioned earlier that cause us all a lot of anxiety. But I think it is still a fantastic moment to be engaged in this adventure of learning things that people didn't know before. And do not worry, there will be support for that. Everybody really wants to see medicine advanced because they're worried about their own health and that of their families and their constituents. And finally, don't forget to have some fun here, because this is not supposed to be one of those things where everybody has got sort of this dreary attitude, I've got to do this work. No, this is actually one of the most amazing gifts you can have, is to work in an area like this with other amazing people and treasure those relationships, because most of the science fun that I've had has been in a team sort of setting, and where your team members have taught you a lot of stuff, enlarged your scientific capabilities, but they've also become your good friends. You're happy to talk about something else, or go for a bike ride, or go to the bar. So nurture those things as well, because that's part of what it's all about. Well, that was a bit of a romp through a lot. But again, I just want to encourage all of you there at the UMBC computational biology group to adopt this same thing. Be a bit daring. That's a good thing. That's what life is about, a daring adventure. And be actually kind of pleased at yourself that you've come up with a path here that's going to be interesting and productive, frustrating at times for sure, but where you have the opportunity to learn things that nobody knew before. And it's hard to imagine anything that one could do with one's life that would be more exciting than that. So I'm going to stop here and see if in a few minutes here, if there are questions, or whether it's possible, Jessuin, to do that. I wasn't quite sure what the format was going to be for that part, or whether I'll be able to hear or not, if people have things they want to ask me.
Yeah, for sure. If anyone has any questions, feel free to come up or type them in the chat. Do any of you guys have any questions? One second. We have a student that has a question.
Great.
I'm very much interested in what you're talking about, the stem cell research. We're talking about the iPSC. How far do you think we have to like converting it to more types, or like, there's an adjacent like organ cell with like, creating like some kind of like storage option, like, where they get like, take out all the organs, like stuff, and then fill up the fillings with like, never mind, I can go on tangent.
We're moving along on that. There are now recipes that you can use to make a lot of different human tissues from an iPS cell. I mentioned we're doing this with neurons, we're doing this with beta cells. We've also done this with cardiomyocytes, and that's kind of spooky. You can get them growing in a dish and they actually start beating right there in the Petri dish, and that means, okay, that's really a cardiomyocyte. We've made vascular smooth muscle cells, we've made endothelial cells. Lots of people are working on the GI tract. And one of the things that gets even more interesting is when you can do that in a way that's not just a single cell type, but you make an organoid. I think what I showed you there that looked kind of like an islet, that was really an organoid because it had multiple cell types. When people make organoids for the brain, that really starts to make you wonder. In fact, there's an ethical question there about how far does one want to go down this road? Is this organoid that I just made starting to develop consciousness? I hope not. So we have to think about those things. And then there's also an effort to try to put these kinds of stem cell derived tissues on a chip and have them interact with each other. So NCATS that I mentioned to you has a whole program of doing stem cells on chips. And the most recent version of that, I think, has 6 different organs on the same chip interacting with each other by shared flow of molecules, which starts to be, a decent representation of a fair amount of human biology, and can be used, for instance, to test toxicity of a drug, or even efficacy of a drug if you've basically loaded it up with organoids that are derived from somebody with the disease. So yeah, the sky's the limit. There's so much that we've already done here, but much more that can't be done. And ultimately, people think we could make transplantable organs this way. And I've seen one example of what looked like a pretty good beating heart that was made from a single person's iPS cells. And maybe at some point will be ready to be reimplanted.
Thank you.
Hmm?
All right, awesome. We have some questions in the chat, if you wouldn't mind answering them. We have a question from Allison. How do you personally balance faith and science? Sorry, this is very general. I support an evolutionary biology class, and some of the concepts I teach seem to conflict with fundamental teachings of my faith, Judaism.
It's a great question. I am a person of faith, but I wasn't raised that way. I became a Christian in my 20s, actually during medical school. And people predicted I was going to have all kinds of terrible conflicts because I was already interested in genetics. I've never really encountered that. I do think there are ways of interpreting scriptures that can lead you to think, wait a minute, that can't possibly fit with what science is teaching us about origins. But oftentimes, those are interpretations of scriptures that reasonable people could say, I don't think that's quite what it meant. Oftentimes, Holy Scriptures of various faiths were not intended to be science books. And when you try to turn them into that, it seems like you're actually stepping away from the original intention. So I wrote a book about this called The Language of God, published way back 18 years ago. If somebody's interested in seeing at least how this one person, who's a person of faith and a scientist, put those things together in a way that actually, I think, feels very harmonious. It's not like I'm apologizing for the fact that there's a conflict and trying to brush it off. I don't think there's a conflict at all. It feels actually very reinforcing, very complementary. You learn more by considering both of those aspects, and you don't have to just push one of them aside on any given day.
Awesome. And we have Victor. What do you see as the greatest challenge in mapping the brain circuits, and how is the Brain Initiative addressing it?
Oh, great question. I think the biggest challenge is the technology that you need to do non-invasive assessment of how these particular circuits are wired and how they function in real time, because you don't want to just measure them statically. You want to see what happens when somebody is carrying out a certain activity. A lot of that work now is getting done by neurosurgeons who are taking care of epilepsy patients who often are admitted to the hospital to try to find out where is that spot in the brain that is setting off the seizure. But that means you got to put electrodes all across the surface of the brain in very fine arrangements. And during the time you're waiting for that seizure to happen, many of those patients are happy to take part in research. And so you can ask them to think about something or do something, and you can really see at a much better level of resolution exactly what's happening there with circuits. You also, of course, can do that in even finer mechanisms in experimental animals using things like optogenetics. I think that's the big push. We have seen an amazing set of advances for human brain science by getting the census of what the cells are in the brain. I mean, I can remember being told, well, there's really only about 12 different cell types in the brain. When you can actually look at single cell biology and you can ask each cell what they're doing, there's more than 3,000 cell types in the brain. Maybe more than that if you really start to get very picky about small differences between cells. It is an incredibly complicated structure and still all differentiated from that one single cell. So yeah, I think that the question is right. That's where the big frontier is, figuring out the circuits, how they work, and then how they don't work in circumstances like schizophrenia or autism or obsessive compulsive disorder or Parkinson's disease, because that's where the big payoff is going to be. When we have a baseline of what normal is, then we can start to understand what goes wrong.
Awesome. And then one more from Megan. How do you suggest we as scientists fight against anti-intellectualism spreading on social media and beyond?
It's a tough one. Social media is, I think, the source of an awful lot of anti-intellectualism, especially in the current climate. It doesn't help to mount a frontal attack because it just escalates everybody's intensity, and pretty soon nobody's really listening anymore. They're just firing off angry statements at each other. I do think scientists have a role to play, though, in trying to draw people back to what the actual facts are. Whether that works very often in a social media setting, yeah, I think it does if it's a place that's not already become completely polarized and full of vitriol. And so it is fine for scientists to jump in there and say, well, hey, wait a minute, let me explain to you what I do and the kind of information that I deal with every day. And let me say why it is that I think something that just got said here isn't actually correct. But do it in a empathetic way where you're not calling the person stupid, which is not going to be very helpful, or evil. You're basically trying to share what you know in a fashion that I think we should all try to do. It is not easy, but I don't think it's right either for us to say it's somebody else's problem. I'm going to go back in the lab. You have a chance to share your expertise. Maybe on social media is not so easy, but go and volunteer to teach a high school class, or be part of other outreaches to the public at your community center. That's the kind of thing we haven't done nearly enough of, and people just aren't really prepared with the tools to evaluate a scientific claim.